Good morning, students. In today's lecture, we will discuss clinical enzymology, that is, the enzymes, their isoenzymes, and their clinical importance. So, the enzymes which are secreted as a result of any abnormality or as a result of any disease condition, then their values are either decreased or increased in plasma or serum. So, on their level, on the studies of their level, we then go in for treatment of that effect. So, first of all, let us see what are isozymes. Isozymes or isoenzymes, they are one and the same thing. Isoenzymes are physically distinct forms of the same enzyme, physically catalyzing the same biochemical reaction. They have distinct physical and chemical properties. There are many types of isoenzymes, that is, true isoenzymes are products of different genes. These genes may be located on different chromosomes. For example, salivary and pancreatic amylase, their genes secrete amylase from different genes. Then alloenzymes, same locus of the gene, having multiple alleles, so produce different isozymes. Then isoforms, due to post-translational modifications, different forms are produced. For example, sialic acid content of alkaline phosphatase, that is, having different concentrations of sialic acid in the alkaline phosphatase chemical composition. Then homomultiple, all subunits are same, and multimers, that is, different subunits, that is, the isoenzyme having different subunits, they are termed as heteromultiple isoenzymes. Then, how can we identify the isoenzymes? First and most important is on the basis of electrophoresis. Then heat stability. Some of them are heat stable, some of them are heat labile. Then inhibitors. Some of the isoenzymes are inhibited by a particular compound, or the other may not be inhibited. Then Km value or substrate specificity, they differ from Km values. Then cofactor requirement, that is, coenzymes and cofactor required for enzymic activity may be different. Then tissue localization. Different tissues have different isoenzymes. For example, your LDH found in muscles and heart muscles and your skeletal muscles, and specific antibodies. They may have specific antibodies. On the basis of these forms or these processes, we can identify various isoenzymes. How isoenzymes help in medicine? That is, medicinal use of the isoenzymes. They are, first of all, most important function of isoenzymes are in diagnosis and prognosis. That is, different isoenzymes are present in different conditions, in different concentrations, in different disease conditions. And when treatment is going on, then the values are again obtained, so the treatment may be modified accordingly. So this way, prognosis is also done on the basis of isoenzyme concentration. So treatment based on diagnosis and prognosis, treatment is carried out, and biochemical assays. So isoenzymes may be carried out, their concentration may be carried out under different conditions, and results obtained, and then treatment and prognosis can be treated or discussed, and then care is taken accordingly. Then diagnosis and prognosis, first of all, the different conditions which we discussed now, so they are individually being discussed. That is, isoenzymes help in diagnosis and prognosis. That is, plasma-specific enzymes are, the plasma is the site of action. That is, blood coagulation enzymes, ferroxidase, etc. These are present in plasma. Then plasma non-specific enzymes, no known function in plasma. Then enzymes of secretion and enzymes of intermediary metabolism. Altered values of these enzymes clinically are significant. Causes of alteration of such enzymes, that is, they may be altered depending upon the causes. That is, first of all is blockage in production while they are being formed. There may be some step which is blocked. Then amount of enzyme production is increased. The reason may be anyone. Their concentration is increased. Then damage of the tissue generating the enzyme. Then when damage of the tissue is there, then concentration of enzyme will be decreased. Then damage to tissues producing enzymes of intermediary metabolism. In this case, leakage will be there, and hence sharp increases observed. For example, LDH. When cells are broken down, then LDH value is increased sharply. And tumor cell growth. Tumor cell growth is also observed with the help of these studies. Now, first example we will discuss is that of lactate dehydrogenase. As you know, lactate dehydrogenase is an enzyme which is responsible oxidation reduction of pyruvate and lactate. When pyruvate is reduced, it will be converted into lactate. It is dependent upon LDH enzyme, which is, the reaction is carried out in the presence of lactate dehydrogenase enzyme, which is dependent upon NADH. And NADH is itself oxidized to NAD, and pyruvate is simultaneously reduced to lactate. And this reaction is reversible, as you all know. So this lactate dehydrogenase level is, normal value is 100 to 200 units per liter. In case of in the presence of that is in RBC within the cells, their values are at least 100 times more than that in plasma. So when there is any injury to RBCs, then the values of LDH will be sharply increased. So upper age limits seen in children. So as compared to adults, LDH values is more in children. Increased levels, increased levels of LDH are observed in hemolytic anemia, that is hemolysis goes on in the blood, then hepatocellular damage, that is liver cells are damaged, muscular dystrophy, carcinomas, and leukemias. Under these conditions, LDH concentration is increased. So, under these conditions, LDH enzymes can be carried out with the value of which indicates which disease or which condition is present within the animal or within human beings. So all of you must be knowing that isozymes of LDH are 5. That is LDH 1, 2, 3, 4, 5. LDH 1 is a tetramer protein. That is LDH enzyme is a tetramer. That is 4 polypeptide chains are there in LDH protein. So LDH 1 has 4 polypeptide chains which are designated as H4. The tissue of origin is heart muscle. Percentage of human serum of LDH1 is 30. Then LDH2 has a composition of H3M1. It is found in RBCs. Its concentration is 35%. Then LDH3, its composition of different polymers, that is polypeptides, is H2M2, that is half of the, that is 2 polymers will be, or 2 polypeptides will be from heart muscle, and 2 polymers or 2 polypeptides are from muscles. So that is liver. So this LDH level, that is LDH3, is having a composition of LDH H2M2. It is obtained from brain. Its concentration is 20% in plasma. LDH4 is found in skeletal muscle and liver. Its composition of the 2 polypeptides is H1M3, and its concentration is 10% of the whole unit or whole content of isozymes. And LDH5, that is last polymer or last enzyme, has 4 polypeptide chains which are obtained from muscles, that is skeletal muscle. Its concentration or its originated from liver and skeletal muscle. Its concentration in plasma is 5%. So these are different LDH isozymes present in the plasma. So this is diagrammatic representation. Then clinical application of LDH. So different units are observed at different levels or different concentrations under various disease conditions. For example, In myocardial infarction, normally LDH1 is more as compared to LDH2. But in myocardial infarction, this concentration of LDH2 and 1 is altered. So in myocardial infarction, LDH2 concentration will be increased or will be higher as compared to LDH1. Again, LDH1 is LDH1 and LDH2 peaks are obtained at 72 hours and stays till 1 week after myocardial infarction. Then muscular dystrophies, in dystrophies of muscles, then LDH5 isozyme is increased. Similarly, in hepatocellular damage, that is liver के damage में LDH5 is increased much. Whereas in case of megaloblastic anemia and renal infarction, LDH1 and LDH2 concentrations are more. Then next comes the enzyme, the isozymes of which are three. This is creatine kinase, or its other name is creatine phosphokinase. So it's composed of two subunits or two polypeptide chains, that is M and B. M represents muscle and B stands for brain. The reaction is carried out, that is creatine, NH2, NH2, NCH3, CH2COO. This is creatine. This is phosphorylated in the presence of kinase and ATP. So you will get, it is phosphorylated, that is nitrogen on left hand side is phosphorylated, that is NPOOO, and the remaining structure remains the same. So this is your phosphocreatine. The enzyme responsible is creatine kinase or creatine phosphokinase, because creatine phosphocreatine kinase or creatine phosphokinase. Its other name is creatine phosphokinase. That is, creatine is converted into phosphocreatine with the help of creatine kinase or creatine phosphokinase. ATP is the energy source with the help of which energy is released and creatine is phosphorylated. Secondly, one more important thing is creatine in case of muscles also help as energy source. So its concentration is higher in muscles. Normal serum level of creatine kinase is for 15 से 100 units per liter in case of males, whereas in case of females it is 10 to 80 units per liter. It is dimer, as you have been told, that is 2 units of M and B are present, 3 isoforms are there, 3 isoenzymes are there, that is CK3, CK2 and CK1, creatine kinase 2, 3 and 1. That is creatine kinase 3 is having 2 polymers or 2 polypeptides of M type, that is, it is obtained from skeletal, and CK1, that is creatine phosphokinase 1, is having 2 subunits obtained from brain. and heart muscle is having M and B, both polypeptides obtained from brain and muscle. So, three types of phosphokinase enzymes or isoenzymes are there. The reaction carried out will be the same. That is, creatine will be converted into phosphocreatine. This is again a reversible reaction. Then, clinical application of creatine kinase. This is muscle, that is heart muscle, in which infarction, myocardial infarction is there. So, the most important application of creatine phosphokinase is in study of myocardial infarction. It is also studied in muscular dystrophy. Creatine muscle, that is MB, that is creatine kinase 2 rises, that is MB. Peaks are obtained in myocardial infarction at 10 to 24 hours, but it returns to normal within 2-3 days, as present in the graph. That is, this is your total creatine kinase. These are hours and these are units of creatine kinase. 0 to 40 unit, 0 to 6 unit, that is total creatine kinase. It returns to zero level in about 60 hours. Then creatine MB, that is the concentration of which is maximum, that is obtained as compared to earlier than other enzymes, that is creatine kinase, LDH, and glutamate dehydrogenase. Though glutamate dehydrogenase is not shown over here in this graph. So, सबसे पहले creatine kinase की वैल्यू राइज करती हैं and they rise at around 15 to 16 hours. They rise maximum and their values are increased at around 10 hours of myocardial infarction. This estimation can lead to the inference of myocardial infarction. This is the enzymic estimation. Then again, besides creatine phosphokinase, myoglobin value is also increased. Besides myoglobin concentration, troponin values are also increased at around 24 hours or 30 hours after myocardial infarction. So, creatine phosphokinase, that is 2, is the most important isoenzyme of creatine phosphokinase, which is studied in myocardial infarction. Then aspartate transaminase, that is GOT, serum, SGOT stands for serum glutamate oxaloacetate transaminase. Serum glutamate oxaloacetate transaminase. So, PLP, that is pyridoxal phosphate, is the coenzyme of this enzyme. Its normal serum level is 5 to 40. जैसा कि अभी जब अपन ग्राफ डिस्कस कर रहे थे, its value is increased in myocardial infarction. ये वाले ग्राफ में, हालांकि this is not being shown over here, but its value is increased in between creatine kinase and LDH. This will come over here. That is in between creatine kinase and LDH. So, normal serum level of SGOT or aspartate transaminase is 5 to 40 units per liter. It is increased in myocardial infarction and moderately increased in liver diseases. Then, besides this transaminase, alanine transaminase is also important for studying clinical enzymology. Its other name is serum SGPT, that is SGPT, serum glutamyl pyruvate transaminase. Then again, pyridoxal phosphate is the coenzyme. Its normal level is also from 5 to 40 units per liter. Its increased level is found in acute hepatitis. GOT हार्ट के लिए है, GPT लिवर के लिए स्पेसिफिक है. दैट इज ध्यान रखने का है, GOT फॉर हार्ट एंड GPT फॉर liver. So increased level of SGPT or alanine transaminase is obtained in acute hepatitis, and moderately increased value is found in chronic liver disease, cirrhosis, hepatitis C. Then comes your next enzyme, that is alkaline phosphatase. It is found in almost all tissues, present in cell membrane, that is ecoenzyme, ectoenzyme. So it hydrolyzes aliphatic, aromatic and heterocyclic compounds. Optimal value or optimum pH of alkaline phosphatase is 9 to 10, and its normal level is from 80 to 125 units per liter. Its concentration or its activity is moderately higher in infective conditions, alcoholic hepatitis, and liver damage. Then very high alkaline phosphatase value is found in obstructive jaundice, that is liver affection, and toxic hepatitis. And maximum value of alkaline phosphatase is observed in bone disease. So it is alkaline phosphatase is most specific, take down, most specific for bone diseases. Then isozymes of this alkaline phosphatase. There are 6 isozymes. These are alpha 1 alkaline phosphatase, alpha 2, that is heat labile alkaline phosphatase, alpha 2 heat stable alkaline phosphatase, pre-beta alkaline phosphatase, gamma alkaline phosphatase, and leukocyte alkaline phosphatase. These are different isoenzymes of alkaline phosphatase. Alpha 1 alkaline phosphatase is increased in obstructive jaundice. Then alpha 2 heat labile alkaline phosphatase is increased, is of liver origin, and alpha 2 heat stable alkaline phosphatase is of placenta origin, that is Reagan isozyme, lungs, liver, and gut. So this is heat stable alkaline phosphatase found in placenta, lungs, liver, and gut. Pre-beta alkaline phosphatase is increased value is found in bone disease, and gamma alkaline phosphatase is increased in ulcerative colitis. So leukocyte alkaline phosphatase value is increased in lymphomas, but its value is found decreased in CML. Then other enzymes as diagnostic markers, these are nucleotide phosphatase, that is 5' nucleotidase. It is marker enzyme for plasma membranes. Ectoenzyme, liver specific. It is liver specific, and its value is increased in biliary obstruction and hepatitis. Then gamma glutamyl transpeptidase. This is nucleotide phosphatase. This is gamma glutamyl transpeptidase. Its normal level is 10 to 30 units per liter. It is increased in infective hepatitis and alcohol abuse. So, its study is most important for alcoholics. That is gamma glutamyl transpeptidase. ध्यान रखो इसका. Its study or its value is studied in case of alcoholics. That is alcohol abuse. This is the most important enzyme. Then acid phosphatase. Normal serum level is 2.5 से 12 units per liter. Optimum pH of acid phosphatase is 4 to 6. Its value is increased in prostate affection, and it helps in follow-up action of treatment also. Then prostate-specific antigen is also studied besides the enzymes. The normal value of this antigen is 1 to 5 microgram per liter. Its value is increased in prostate, and it is used as a tumor marker. Then cholinesterase. Its true cholinesterase is found in RBCs, and pseudocholinesterase is found in liver. Now, true cholinesterase found in RBCs and nerve endings, cholinesterase level is directly proportional to the reticulocyte count. Its level is increased as a result of organophosphorus poisoning. And pseudocholinesterase is found in liver cell, and it is studied in diagnosis of hepatitis. Then glucose 6-phosphate dehydrogenase. This is an important enzyme of hexose monophosphate shunt, that is pentose phosphate pathway. It is dimer, that is 2 identical subunits are there of this enzyme, and this enzyme is most important in production of NADPH coenzyme. As you all know, NADPH is required for maintaining RBC integrity. Normal serum level of glucose 6-phosphate dehydrogenase is 6 to 12 units per liter. Its importance lies in drug-induced hemolytic anemia. Then amylase. Amylase is the enzyme which splits starch to maltose. It is found in pancreas and salivary glands. It is activated by chloride ions and calcium ions. Its serum level is 50 to 120 units per liter. It is increased to 100 times in acute pancreatitis. So in case of pancreas affection, its value can be increased 1000 times. It is moderately increased in chronic pancreatitis, mumps, and obstruction of pancreatic duct. Then lipase. It is also secreted by pancreas. Its levels are increased in acute pancreatitis. This enzyme is more specific than amylase studies and gold standard for diagnostic pancreatitis. Then last enzyme is enolase. That is, this enzyme is neuron specific enzyme. This is tumor marker for cancers associated with neuroendocrine origin, lung, neuroblastoma, and pheochromocytoma. Thank you for this lecture. I hope you will mark them up. Thank you. So, one thing I may substantiate it. Enzyme pattern in different diseases are hepatic enzymes क्या क्या हैं different diseases में, that is liver में कौन कौन से enzyme study कर सकते हैं, that is alanine transaminase, that is GPT, aspartate transaminase, that is GOT, alkaline phosphatase, then nucleotide, that is nucleotidase, glutamyl transaminase, GGT. Then cardiac markers are enzymatic markers, LDH, GOT, creatine phosphokinase, and non-enzymic markers are troponin, T and I, myoglobin, then blood nitrogen, and copeptine. Then muscle diseases में creatine kinase MM, that is creatine kinase 1, AST, that is your GOT, aldolase. These are the enzymes to be studied in muscle diseases. And bone marrow, that is alkaline phosphatase. Prostate cancer के लिए you can study prostate specific antigen and acid phosphatase. So therapeutic use of various enzymes, that is various enzymes are used as drugs, that is in treatment. That is asparaginase, it is treated widely. Then streptokinase and urokinase, it lyses intravascular clot, that is myocardial infarction के बाद. Streptodornase, that is DNase applied locally. Pancreatin, pancreas insufficiency, papain, it is used as an anti-inflammatory. Alpha-1 antitrypsin, this is used in emphysema. And enzymes in diagnostic assays. So these are used for routine diagnostic purposes, that is glucose oxidase for glucose, urease is studied for urea estimation, Taq polymerase is studied in PCR, polymerase chain reaction, reverse transcriptase is studied in, it is utilized in recombinant DNA technology, and horseradish peroxidase is utilized in ELISA, that is enzyme-linked sorbent assay. So this is regarding various aspects of enzymology. So thank you. आज
स्टॉप करिए ना सर,
तो है ही नहीं है, कहाँ गया?